Plant frequency, an overview. Frequency reflects the likelihood of encountering a plant species at any location in an area of interest or landscape. The frequency is defined as the number of times a plant species is present in a given number of uniformly sized quadrats that are placed repeatedly across a stand of vegetation. Frequency measurements focus on the presence of species within quadrats, and this is an important distinction from density, in which we count the number of individuals in the quadrats. It is very sensitive to changes in the size of the quadrat. That's why quadrat size is part of the definition. So it's important to remember these two aspects of the definition for frequency. It depends on presence versus absence, and it is strongly influenced by quadrat size. Frequency data are used commonly to monitor vegetation changes. It is very sensitive to changes in the distribution of plants as well as the number of plants. And we often use frequency to assess vegetation trends because it can be used to compare vegetation changes at one site over time, or to compare two different sites that support similar types of vegetation. Frequency is calculated as the proportion of the number of plots in which a species occurs relative to the total number of plots that are examined. The frequency calculation is quite straightforward, and we always report frequency as a percentage, so this ratio is multiplied by 100 to turn it into a percentage. So let's look at an example. We have a graphic here of a macro plot with three species. There's a thistle plant, there is a pink daisy, and also grass plants. We've also located three quadrats in this macro plot and then calculated the frequency of each one of the plants. The thistle plant occurs in one quadrat out of the three, and so that calculates out one plot divided by three plots multiplied by 100 equals 33% frequency. We follow the same procedure for the daisy. It's located in 2 out of 3 of the plots, which translates to 67% frequency. And for the grass, we have grass plants that are present in each of the 3 plots, which calculates to 100% frequency. It's important to note that the fact that there are multiple grass plants in this quadrat and in this quadrat does not influence the frequency calculation or measurement, because we're only looking for presence, and the number of individuals that occur in each quadrat is not of importance. Simply having one plant in the quadrat will count as presence. Now let's consider the relationship between frequency and density. In this schematic, we have 2 species, a dandelion and a daisy, and we're going to calculate the density and frequency of both species using these 3 quadrats. The dandelion is present in 2 of the 3 quadrats, and there's 1 plant here and 3 plants here. So we have a total of 4 plants in 3 plots, which calculates to 1.3 plants per plot. The daisy has the same density. There are 4 plants in this plot and none in the other 2, so that still calculates out to 1.3 plants per plot. We can see that the frequency is different because the dandelion is present in 2 of 3 plots for 67% frequency, yet the daisy is found in only 1. It's present in only 1 of the 3 plots for 33%. Let's next consider the relationship of frequency to the spatial distribution. In this pair of diagrams, we see another schematic graphic of vegetation. On the left, the vegetation is patchily distributed, so the plants are clumped. There's a lot of open space. In the right, we have the plants are more evenly distributed, more homogeneously distributed throughout the area. Now, because this is a graphic and we're able to manipulate the items in the graphic, I can tell you that the density of plants, the number of plants in this diagram on the left, is identical to the number of plants in the diagram on the right. In addition, we kept the same items in both diagrams, and so the actual cover is also the same between the two diagrams. The only difference is their spatial distribution. We've located 9 quadrats in each one of the diagrams, and we can see that out of the 9 diagrams, the 3 quadrats that are in blue have plants present in them. And that translates to 33% frequency. In the diagram on the right, the more homogeneously, evenly distributed plants, we have plants present in 8 out of 9 of the quadrats, which calculates to 89% frequency. So again, the only reason for the change in these frequency measurements is the spatial distribution of the plants. There are numerous advantages to using frequency measurements. First of all, they are objective and repeatable. Because they're based simply on presence, there's not much room for bias from the observers, and we do tend to have repeatable measurements taken by different observers. It's relatively rapid to take these kinds of measurements, and it's relatively simple. It requires some training, but not an extensive amount of training, as long as your plant identification skills are good. Frequency is relatively stable to environmental fluctuations. And so this means fluctuations that may occur between seasons or between years. It's also stable to the removal of biomass by grazing animals. There's no need to distinguish individuals, so that helps to save time and is one of the reasons that it's a relatively rapid measure to take. It's a sensitive indicator of changes and trends, and it is affected by both the density and the plant dispersion. This is an advantage because when those characteristics change, we want to be able to detect it. But this is also a disadvantage. And one of the reasons we consider this sensitivity a disadvantage is that if a change in frequency is detected, we don't necessarily know whether the change was due to a change in the density of the species or a change in the spatial distribution of the species, or both density and spatial distribution. The only way we could know the reason that frequency changes is if we collect other information at the same time. It is sensitive to quadrat size, and that's an important feature because sometimes we may need to use different size quadrats if there is a change in the vegetation and we want to continue monitoring the plants. We may need to make some adjustment to quadrat size, and in that case that takes a special approach to documenting that we've changed quadrat sizes in order to be able to use those data for comparative purposes. As I just said, different size quadrats may be needed if there's a change over time, or for different species, we may need different size quadrats. Also, frequency can be very sensitive to seedling numbers. If there is a large recruitment event or a large seedling emergence event, and we're simply measuring the presence of that species, having a lot of seedlings can artificially increase the frequency that is measured for a particular species, even though those species may not live through the year or long enough, and so there may be an artificial sense that the frequency has increased when over the long run, if those individuals die, then the frequency of the population hasn't really increased. And finally, frequency can be difficult to visualize because it's not an absolute attribute of the plants. We can say for sure that there is a specific density of plants. That is a parameter that can be measured, and it is an attribute of the plants in the environment. The same can be said for cover or for biomass. But because frequency measurements depend on the size of the quadrat, and they change with the size of the quadrat, it is not an absolute measure of the plant population. It's not independent of quadrat size. There are important ground rules that need to be established when we're measuring frequency. First, what do we count as being in or out of the quadrat? We also need to determine the level of identification that is to be used, and also the number of samples that are used. Sample size is always an important consideration, and so how many samples is going to be enough. And finally, the quadrat size. Determining the appropriate quadrat size is a very important consideration that needs to be determined. All of these considerations are driven by management objectives and are determined on a case-by-case basis. Now let's consider the criteria that we use for counting a plant as inside the quadrat. Normally we use the principle of rooted frequency. If a plant has its roots, if it is rooted inside the quadrat, we would count that in the quadrat. And this works not only for single-stemmed plants, but also for mat forming plants, such as this grass mat that we see here. It doesn't matter how much of the plant is in or out, as long as part of the plant is rooted inside the quadrat, that counts. Now, sometimes we can have plants that are stoloniferous plants where the main plant is rooted outside of the quadrat. But it can have ramets on the stolons, and if those ramets are rooted in the plot, they would count. Note that this particular ramet does not have roots established in the plant, and the main plant is not located in the quadrat. And so since it does not have any rooted part in the quadrat, we would consider this plant as outside of the quadrat. It doesn't matter that the ramet is here that the stolon is here or that there is overhanging canopy. Rooted frequency is the standard that we use for counting plants as in. That said, there is a special adaptation that we use on occasion in which we do count canopy frequency. This is usually employed only when we are concerned with being able to detect large plants that are dominant in the landscape that might otherwise be missed, because we would need a extraordinarily large quadrat in order to encounter that plant inside the quadrat. So here we see an example with a tree that has canopy that is hanging over into the quadrat, and so we would count this one as being in the quadrat. In the diagram at the bottom, we would not count either one of these trees as being in the plot because the canopy is not overhanging. Now it's important to note that we only use this for certain plants that are important for us to be able to detect, and we need to justify using canopy frequency for those plants. It's also important to clearly document when this approach is used and explicitly which species it's used for. Next, let's consider the level of identification. Usually, when we're talking about taxonomic level, oftentimes we focus on species, whether the identity of a species, whether it's present or absent in the quadrat or plot. Depending on our management objectives, it may not be necessary to identify plants to species, especially for those genera where members of the same genus are very difficult to determine to the nearest species, and also where they respond similarly to management and they fill a similar role in the environment. So it depends on your management objectives whether you need to identify to the nearest species, identify to the genus, or possibly identified to just the functional group, whether it's a grass or a forb or a woody plant, etc. The age class also needs to be determined. Oftentimes we will count any individual, if it's present, whether it's a seedling, a sapling, or a mature plant, as being present in the plant, in the quadrat. Sometimes we are interested in knowing whether it's a seedling, sapling, or a mature plant, and so we can quantify that information. Sometimes we're interested in whether the plants are reproductive or have become senescent. And sometimes it's not important for us to make any kind of age distinction or stage, reproductive stage class distinction. It's important to consider this, because when we are collecting data, if we write down that we have a seedling, or if we're making the distinction of, say, seedlings versus saplings versus mature plants, it's very difficult to lump those back together in our when we're doing the analysis, unless we take special care when we are collecting data to collect the data on a quadrat by quadrat or plot by plot basis. So it's very important to determine how important it is for us to know this information before we start to collect it. So let me reiterate that a little bit. If we had a quadrat that had a seedling and a mature plant of the same species, if we weren't making the distinction between seedlings and mature plants, we would simply say that plant was present. However, if we were making the distinction, we would mark that the seedling was present. We would also mark that a mature plant was present. And it would be very difficult in analysis to then lump seedlings and mature plants together to get an accurate measurement of the frequency of that particular species. Finally, with annual plants, we always need to make a determination of whether we're going to count annuals, or if we do count annuals, do we identify them to species, or do we just count them, do we lump them together as annuals? A lot of times annuals are fairly ephemeral and they only live a short period of time. If we want to note their presence, do they have to be alive when we see them? Or if we're capable of determining the identity of the plant, even though the plant has died, do we count it? Sometimes people will only count perennials because of the ephemeral nature of annual plants. And sometimes what people will do, depending on the situation, is just lump all annual grasses together and lump all annual forbs together. Again, this is very situation specific, depends on your management objectives, and it depends on the environment that you're working in. Obviously, if you're working in the California annual grassland, it would not make sense at all to simply lump all annuals together and count them as a functional group. Let's consider the importance of sample size and design. Usually, in order to detect changes, we need at least 100 to 200 quadrats in an environment, and sometimes more would be needed in order to detect small changes. In general, the quadrats are arranged in a macro plot. So we can consider the macro plot here as being our area of interest. And they are systematically placed throughout the macro plot or the area of interest. The spacing is important because this does affect the independence of measurements. Now, I just said that they were systematically arranged, which means that we usually violate the assumption of independence when we use systematic arrangement of sampling units. However, depending on the size of the plants and the plant patches, if the spacing is determined so that we are sure that one quadrat does not measure the same plant or the same plant patch as another quadrat, then that helps us with the assumption that the measurements that we're taking for frequency are independent. And finally, let's address the importance of quadrat size. What we see here are three diagrams, again, of the grass and the daisy and the thistle, and the three diagrams are identical in terms of the plants in the diagrams. The only thing that's different is the quadrat size. We have 4 quadrats, they're located in the same place, but the quadrat size has changed. And if we look at the effect of this on the frequency measurements of the pink daisy, we can see that the frequency increases as quadrat size increases. And that's because as we lay down a larger size quadrat, it's going to be able to encompass a larger part of the environment, and it's going to capture more plants in the quadrats. So this illustrates the direct effect or the direct influence of quadrat size on frequency measurements. There's no difference between the actual vegetation community here, but the measurement of the pink daisy frequency changes with changing the quadrat size. There are several guidelines that we use that pertain to determining the appropriate quadrat size. In general, the greatest sensitivity to be able to detect changes in frequency are when we have frequency measurements that are between 20 to 80%. And we generally try to target 30% to 70% frequency for most species. So we want to be able to choose quadrat sizes that are going to give us measurements in these ranges. And this is because of statistics and of our limitations to being able to detect changes in frequency at very low frequencies or very high frequencies. In general, if we find that our frequency measurements are below 10%, we need to increase the plot size that we use. On the other hand, if frequency measurements are greater than 90%, we need to decrease the plot sizes. We select the plot size that works best for most species, and I need to reiterate, when I say plot, I'm also meaning quadrat. I'm using these terms interchangeably. We use nested quadrats or nested plots when one size won't work for all species. And a discussion of how to select the appropriate quadrat size and how to determine when to use nested plots and how to do the calculations using nested plots is the subject of the subsequent lesson.